All right, so we're going to talk a little bit today about parasitic protozoa. We mentioned that we'll be looking at three different classes of parasites in this course: protozoa, helminths, and then some arthropods. Today, we're going to focus on some of these parasitic protozoa and how they can potentially cause human disease. We've spoken briefly about the occurrence of parasitic diseases, and so I just wanted to remind you that we typically see these parasitic diseases in places that are typically underdeveloped, rural areas, places that are overly crowded. And some of the global distribution of parasitic diseases, of course, if you can look at this map here, we have the leishmaniasis are kind of prevalent over in the African, Asian regions. Trypanosomiasis are ones that we see in the Americas, both North America and South America. I think you guys had an extension with T. cruzi. So one of the weeks past. We see schistosomiasis over in the Europe area. We see malaria very heavy in the African continent. So these are some of the representative global distributions of parasitic diseases. We're going to take a deeper dive into this conversation today. Real quick, though, we talked about some routes of transmission for parasitic diseases. One of the most common, and particularly with parasitic protozoa, is the fecal oral route, right? So some way or another, an infected individual ingests, okay, some contaminated foods or water, and can become infected with things like entamoeba, giardia, toxoplasma species. We'll look at some of the fallout of that, some clinical manifestations of things like entamoeba infections, giardia. These are typically going to be intestinal discomfort type clinical symptoms. So fecal oral route is one of the most common routes of transmission, particularly for these parasitic protozoa. We also see vector-borne routes of transmission where we have an arthropod or some other mechanism that serves as a vector or a vehicle for transmitting these organisms, right? So we see that with Plasmodium species, Leishmania, Trypanosoma. These all are going to be transmitted by way of a vector. For Plasmodium, it's the mosquito, the Anopheles mosquito, I think. And for Leishmania, it's, I want to say the phlebotomine sandfly, maybe. But some type of arthropod is transmitting these infectious organisms. Sexual contact also cannot be omitted as a route of transmitting parasites. Trichomonas, we see that being transmitted with the clinical disease trichomoniasis. And then contact, all right? So these organisms can penetrate the skin, all right? We'll look at some of that when we talk about our schistosomas, schistosomiasis, and the different types of organisms that help to create these types of direct contact transmissions, all right? So real quick, again, we have three major groups or classes of parasitic organisms. We have the protozoans, we have the helminths, and then we have these arthropods, right? Let's look today at, let's recap this idea of classification. We know that there's a five kingdom, we have the five kingdom classification where we know that there's life, and at the domain level, we have our three domains of life: eukarya, prokarya, and archaea, right? And after this domain level of classification, we've got kingdoms, right? So there are five kingdoms that you are probably familiar with, but you do need to review this information if you've forgotten it as we start identifying these new classes of parasites, all right? So we've got plantae, fungi, animalia, protista, and prokarya. This is where we find our bacteria, right? So we're going to look at some types of protista today. This is where we find our protozoan organisms, right? And so the reason I put this slide in here is so that you can now start to recall all of these identifying characteristics of protist. Okay. What do you recall about protists, just in your general biology? What are some general characteristics of protists? Anyone? Anyone? Any type of classification factors? Any properties? No one? Okay. So let's look at some of these characteristics of protists that you probably should be familiar with. So these are a unique class of animal-like organisms. In terms of the domain, they are eukaryotic. All right. They're obviously microscopic and they are unicellular. Go ahead and jot some of these characteristics down. You do need to know them. These are just some general characteristics of protozoan organisms. So they're eukaryotic, they're unicellular, they typically lack a cell wall. They're going to move via these pseudopodia, flagella, cilia, different structural appendages and attachments that facilitate movement. Okay. We have, like, for example, right here, this would be an amoeba. We see the presence of these pseudopodia, which are going to facilitate motion, motility, movement, locomotion. Okay, we see the presence of a flagella here on this organism. This flagella also aids in movement. We've got cilia over here on this organism. So these are all examples of protozoans, all right, and their structural arrangements, all right. Characteristically, they primarily reproduce by way of asexual reproduction. All right. You should remember that asexual reproduction only requires a single individual to occur. One example of asexual reproduction is binary fission. That's typically how a lot of them reproduce. It's very similar to cloning. All of the newly produced offsprings will be an identical replica, if you will, of the parent cell. All right, so there is the genetic diversity doesn't happen with this mode of reproduction. Whereas with sexual reproduction, you'll recall that the offsprings that are created during sexual reproduction are hybrids. Okay, hybrid organisms or hybrid individuals, which basically means they have shared genetic content from both mom and dad. Okay. Parasitic protozoa can be either free living or obviously parasitic. So protozoans, rather, let me say this, parasitic protozoans in general can be either free living or parasitic. Mostly we're going to find them inhabiting water or soil. Okay, they're symbiotic in and on animals, and some of them can cause human disease, which is why we care about them in this course. We're going to look at a few protozoan organisms that do cause problems in humans. All right, so, you know, generally speaking, you should be able to rattle off a few characteristics, or if you, whenever you hear the term protozoan, certain characteristics should come to mind from a classification standpoint and being able to identify what types of organisms they are and features that they exhibit. Okay, eukaryotic, unicellular. They move by way of the pseudopodia, flagella, other structures. They're reproducing via asexual reproduction. We're particularly interested in those that are parasitic. We can find them again inhabiting water and soil. And unfortunately, some of those do cause human disease. And we're going to take a look at some of those today. So just from a point of reference, nearly all humans have been infected with a protozoa at some point in their life. All right. Some of these species are commensals while others are pathogenic. So we've talked about these terms that we're looking at here. Who remembers what is a commensal? What we talked about, some relationships between organisms. We talked about symbiosis, commensalism, parasitism. What is a commensal relationship?
It's one that one specific benefits and the other is not hip or harm.
harmed. Right. So parasitic infections can range from mild to life-threatening. We will look at a few common parasitic pathogens. Toxoplasma gondii, you've probably heard about before. Cryptosporidium, I think we mentioned that early in our discussions, and Giardia. These are common protozoan pathogens that can cause problems. All right. So one way that we can classify protozoans are based on their method of movement. Okay. We have amoebas, we have the ciliates, and then we have the flagellates. All right. These amoeba are these flat jelly-like organisms, and they have pseudopodia. All right. These pseudopodia are basically these fat little flabs that we call false feet, right, that help them to move. All right. So structurally, if I showed you some images, you should be able to classify these organisms, okay, based on the structures that are present to facilitate locomotion. All right. So we can identify protozoa as amoeba based on the presence of these pseudopodia here. We can identify them as ciliates, okay. These ciliates are characterized by the presence of cilia, okay? These small, thin, hair-like projections that come off of the body of these organisms. These are the ciliates, okay? And then we have the flagellates. Flagellates are those that are going to have this nice flagellum, okay? So we are saying that we can classify protozoans based on various features or characteristics. And the first one is based on how they move. Okay. Parasitic protozoan that enters the humans through ingestion. Okay. We talked about ingestion, that fecal oral route, ingestion. Okay. These organisms, those that are entering the human via ingestion, are going to have two morphological forms. I want you to understand this. Okay. This is important, right? We're talking about parasitic protozoan. We're specifically talking about those that enter the humans via ingestion. So you've consumed some contaminated food or water, all right? These organisms have two morphological forms, okay? They have a trophozoite form, and then they have a cyst form, okay? Two morphological arrangements of these parasitic protozoans that enter by way of ingestion. Okay. So this trophozoite phase is going to be the feeding and reproductive stage that lives within the host. Okay. While it's inside of the host, it's going to undergo a process known as encystment. Okay. During encystment, after encystment is going to allow this encysted organism to basically move throughout the individual before being released in feces. Okay. Then the cyst form of the organism is the infective form that now survives out in the environment. Okay. And it is going to be these cysts that somehow or another contaminates the food or water that we end up drinking. Okay. And once it's ingested, the process of encystment occurs once inside of the individual. The trophozoite basically is released, and it starts to repeat that cycle. Okay, so we have a repeating cycle of trophozoite and cyst form. The trophozoite phase, again, is going to be this feeding reproductive stage that's happening inside of the host. It's going to undergo encystment. Cysts are going to form and be released from us during defecation in our feces. This is what makes its way out into the environment. It ultimately will contaminate our food or water source, right? Animals, cattle. And encystment happens once it is re-ingested and the trophozoite phase repeats. All right. So the two morphological forms right here, if we were to look under the microscope at these two different stages of development, all right, we have a trophozoite arrangement, and then we have this cyst right here. All right. This is what the trophozoite would look like under the microscope, and this is what it would look like during this cyst stage. All right. And so again, you know, we're not expecting you to be experts at the end of this, but we do want you to have some basic understanding of what's happening in the life cycles of these organisms. And so this trophozoite stage, again, is just, it's what we'll kind of refer to as the growing stage, right? This is the stage for which it has infected the host. It's active, it is reproducing, okay? And this is the phase for which it is infecting the host, okay? Before it leaves the host in the feces, right, it's going to undergo encystment, and a cyst is going to form around it. All right. The cyst stage is how it persists out in the environment. All right. And so like if we, I think we talked a little bit earlier about diagnosis of parasitic infections. One of the ways we diagnose parasitic protozoan infections is collecting stool samples. And so we will be looking for these cysts in the stool. Okay. We can also identify trophozoites in an individual who has like diarrhea or something where it's not a formed stool. We can probably find a trophozoite stage also. All right, but we usually find these cysts out in formed solid stools. All right. So let's look at the amoeba. All right, amoeba are one of our examples of protozoans. All right, again, we know that protozoans are unicellular. Okay, they don't have any defined shape. They just kind of look like a flat blob. They move and acquire food via pseudopodia. Okay, these are all pseudopodia right here. These little extended appendages that kind of look like fingers. All right, these are pseudopodia. All right, this aids in movement. Amoeba can be found in water sources and soil, pretty much anywhere worldwide. All right, for the most part, they're commensal. Few of them can cause disease, which are the couple that we're going to look at here today. Entamoeba and Naegleria species can cause human disease. All right, so these are amoeba. Let's look at the Entamoeba species. All right. The one organism here that causes human disease in particular is Entamoeba histolytica. This is the genus and species. This organism is spread via a fecal-oral route of transmission. Okay, it is detected in the feces of infected patients, right? So in order to have a definitive diagnosis of an infection with Entamoeba histolytica, we've got to be able to collect the stool. All right. They're actively motile trophozoites, can be seen in freshly passed stools. Okay, so some of them, these trophozoite phases, like some of them appear in showers, and we may have to collect several stools before we can actually get some actively motile trophozoites being present. And again, those hard, more formed stools that may be out in the environment for whatever reason, typically we're only going to find cysts in those more formed stools. Okay. So Entamoeba histolytica, so this life cycle, let's look at this pathogenesis cycle. All right, so basically you're ingesting the cyst in either some contaminated food or water. All right, so the water hasn't been properly processed, or your foods are still undercooked and they are contaminated with cysts that of this Entamoeba. All right, so the cysts are going to pass into the stomach, right? And the gastric acids are basically going to stimulate the release or the excistement, okay, the release of these pathogenic trophozoites into the intestines. All right. The trophozoites are going to continue to divide and cause a lot of damage in the intestine. All right. A lot of necrosis, okay, tissue destruction in the large intestine. Okay. And this tissue destruction or necrosis is going to be due to the presence of cytotoxins. All right. Again, either the trophozoites or the cysts can be ultimately released in feces and making its way out into the environment, and the cycle will continue. Okay. I would like for you to be able to give me a general description of what happens during pathogenesis with Entamoeba histolytica. You're basically ingesting some kind of contaminated food or water. The cysts are going to make its way into your stomach during the process of digestion, right? Excistement occurs. That cyst basically is removed or released, and trophozoites are now active in your stomach, moving into your intestines. Those trophozoites are basically going to cause necrosis. Okay, a lot of damage in the intestines. The cytotoxins that are released are going to cause tissue destruction. And again, in order for us to be able to make this definitive diagnosis, we would need to identify either those trophozoites or cysts present in feces. All right. So this Entamoeba histolytica life cycle is a very simple life cycle that cycles between the trophozoite phase in the human and the cyst phase out in the environment. So I'd like for you to describe what's happening with excistation and encystation, encystment and excistation. Okay. So the formation of the cyst and then the release of the trophozoite in the human. Okay. The trophozoite phase, again, is going to be that actively reproducing phase, feeding phase. It's motile. This is inside of the host. The cyst phase is kind of the infective phase. It's going to be resistant, and it's going to be passed out during defecation. Okay. What questions do you have about this? It's a very simple schematic that represents what's happening during the Entamoeba histolytica life cycle. I would like for you to be able to describe what's happening during this life cycle for me. All right. And what leads to the pathogenesis. Okay. How is this organism a human pathogen? Okay. Clinical symptoms of Entamoeba histolytica, again, amoebiasis. Amoebiasis is the disease, okay? It's characterized by tissue destruction in the large intestine. Obviously, that's going to be coupled with abdominal pain, cramping, colitis with a little diarrhea, dysentery, which is going to be like a watery diarrhea, bloody stools. That's what dysentery is. All right. So basically, it's characterized by intestinal discomfort. Okay, tissue destruction in the large intestines. Okay. So intestinal discomfort, inflammation of the intestines, basically. The carrier state. You can these symptoms can be present depending on the individual, and we talked about factors that can influence the clinical symptoms of an infection. Some individuals can remain asymptomatic. Okay. Some organisms may reproduce, and cysts may be passed in the stools with absolutely no clinical symptoms, right? That can be due to, you're ingesting a strain that has a low virulence. You ingested just a low inoculum, so not enough of the actual organism to cause significant damage. All right. You are a healthy individual with an intact immune system. These are all reasons for why an individual may be asymptomatic and not show clinical signs of amoebiasis. Okay. So I would like for you to be able to understand this infection. Okay. Entamoeba histolytica can definitely have some very serious clinical symptoms, this amoebiasis, or an individual can present in this carrier state where they're asymptomatic. And the only way we know that you're infected is because you're passing these stools, these cysts out in your stools, but you do not exhibit any clinical symptoms. Okay. And reasons for that, again, you've ingested a strain with very low virulence. So your immune system is not responding too much to it, or it's not causing many problems in your intestinal tract. Or the low inoculum, okay, you just only had a little bit. So you kind of, you're not really going to have much response to it. And then because you are healthy, you have an intact immune system, we can rid your body of it pretty easily, and you will not manifest any symptoms of the infection. I have a really quick video here that runs down the infection.
Grammar and spelling are important, but if you want to write essays that inspire.
Oops. Hold on, guys. All right, so I really like this illustration right here because it shows you each stage of the Entamoeba histolytica life cycle, right? Let me get the entire schematic back up here so we can look at it. All right. So we're seeing right here that these cysts are going to be released, and the trophozoites or the cyst will eventually be re-ingested, okay, into the human host. Excisation is going to occur once that you ingest those cysts, and the trophozoite stage will resume. All right. During that time, replication, multiplication is going to take place of these trophozoites, and the new cyst will be forming as it moves through your digestive tract, and these cysts will ultimately be released from the host in feces. All right, this cyst is going to be now our diagnostic mechanism. All right, in our stool samples we're collecting, we're able to identify the presence of cyst in these formed stools. All right, if we happen to catch the patient during a diarrheal episode, we might be able to identify or isolate trophozoites. All right, but typically we're having our cyst at the diagnostic stage. All right, so I like this schematic right here. All right, so let's keep going. Oops. Share. All right, so those were the amoebas. Let's look real quick at the flagellates. All right, so we're still classifying these protozoan based on how they move, the flagellates. These are small single-celled organisms that have this leaf-like appearance. All right, so again, if I showed you some micrographs or pictures of these organisms, I would like for you to be able to identify them. Okay, this leaf-like appearance typically contains at least one flagellum. Okay, this flagellum is aiding in movement. The number and arrangement of flagellums help to identify the specific species. These can be found in streams, lakes, fresh waters, basically mountain resorts and so forth. All right, examples of flagellates are our Trypanosoma, Leishmania, and Giardia. This picture right here is a picture of Giardia. This is a very common parasitic protozoa. So you will see these images quite often as you're doing research and working on your extension assignments for parasitology. You'll see Giardia quite frequently. All right, this structure is very prominent. All right, these structures that are here are quite prominent. You see these multiple flagella. I can't tell you what all the physiological structures are, but I do know that when I see an image that looks anything remotely like this, I know that it is a Giardia organism. Okay. Giardia lamblia, write this one down. All right, this is definitely a human pathogen. All right, again, we have that same prototypical cyst and trophozoite phase that's detected in fecal specimens from an infected patient. Acquired, we talked about that fecal oral route of transmission, which basically means there's some type of consumption of inadequately treated contaminated water or the ingestion of contaminated undercooked food. All right, we definitely have specific recommendations for cooking our foods to a proper temperature so as to get rid of any potential food pathogens that are present. All right, this is a nice little micrograph down here of what these organisms look like under the microscope. We see the presence of these multiple flagella. Okay, so Giardia lamblia is certainly an important parasitic protozoan pathogen of humans. Okay, it's also an intestinal parasite. All right, these are our flagellated parasitic microorganisms. They're going to basically colonize our intestinal tract. Okay, they're going to cause very characteristic symptoms like diarrhea, cramping, and so forth. All right, so Giardia lamblia, let's look at the pathogenesis. Ingestion of the cyst is pretty typical. All right, ingestion of the cyst, excistation occurs as those gastric acids are active in the stomach. All right, we're going to release those trophozoites into the intestine. They're going to continue to replicate or multiply, and it's going to cause necrosis in the intestine. Very similar to what we saw with amoebiasis. Okay. This is our trophozoite looking right here. This is what a trophozoite looks like the trophozoite organism looks like under the microscope. And this is what the cyst stage looks like under the microscope. All right. Giardia lamblia. Okay. Very similar life cycle again to that of the amoebiasis. Okay. So we're ingesting some type of contaminated food or drink. It's contaminated with cyst, the infective form. Okay. Do make sure you have that in your notes and you understand the distinction here. All right. So we ingest the cyst, excistation occurs, replication or multiplication of these organisms are occurring now, the trophozoites. All right. And then the trophozoites again are moving through our digestive tract. They're going from the small intestines into the large intestines. Encystment takes place. These cysts are released in our feces and the cycle repeats itself. Okay. All right. Symptoms of Giardia lamblia infection. All right. Giardiasis is the disease. Giardiasis is the disease. This is one of these ones where you're going to have an upset stomach really quick. All right. Infection with Giardia has sudden onset. You're basically going to get some watery diarrhea. It's going to smell awful. All right. It's going to be coupled with abdominal cramps, flatulence, you're passing gas, you're gassy, you're crampy, and you're pretty much gonna be on the toilet pretty quickly. Some individuals, about half of the individuals who are infected can remain asymptomatic, okay, for reasons we spoke of earlier, right? I do want you to understand this sort of a connection with being asymptomatic or not, right? Individuals with intact immune system, again, small inoculum or strain, a strain with the low virulence or low inoculum size, or, you know, just having a pretty good immune system. You can pass the infection with as an asymptomatic carrier. All right. All right. Lastly, the ciliates. All right. The ciliates are going to contain these hair-like projections that are called cilia. They also aid in motility or locomotion. They are going to be present in a very large number. All right. Again, we have this asexual mode of reproduction here. We can find these ciliates out in the environment in water. So lakes, ponds, ocean. All right. We can also find them in the soil. All right, so these ciliates, so we have our amoebas, we have our flatulates, and then we have our ciliates. These are all types of parasitic protozoa based on how they move. Okay, ciliates, Blantidium coli. We call it B. coli, just like E. coli, but this is B. coli. This is the only ciliate that is a human pathogen. Write that down. All right. This is the only ciliate, all right, that is a human pathogen. Okay. Infection is associated with contamination of water supplies with pig feces. Okay. So this one is spread via person-to-person contact. B. coli, Blantidium coli. All right. So what does B. coli pathogenesis look like? Again, ingestion of these infectious cysts. These cysts are going to release the trophozoites. They're going to invade the mucosal lining of the intestine. Reproduction is going to take place. The same thing that we've seen with both the Giardia and amoeba. All right. Reproduction occurs and encystment occurs or follows, and these cysts are passed with feces. See if we can get this other video to work here.
So let's say you have to read an incredibly long email from your boss that you have to finish before the big meeting starts in 10 minutes.
So what I'd like to do is start, you have a table in your book, and what I've done just to make this a little easier is we're just going to follow the table straight down, starting with the first, forget it. So we'll put it, talked about two back
Oops. Hold on, guys.
Material agents of dysentery.
Hold on, guys. Share. Okay, there we go.
See if you can think about what those are. We talked about enteroinvasive E. coli, and we talked about Shigella dysenteriae. And this is amoebic dysentery. Now, one of the things I want to show you might be on a pathology type report from a patient with entamoeba. You get these flask-shaped lesions, all right, from entamoeba histolytica, and you can get extension into other organs like the liver, for example. When you get extension into the liver, this is a very severe complication from entamoeba. And one of the things you can also see with an entamoeba infection is pseudoappendicitis. So you can also get a pseudoappendicitis-like disease, like we talked about with another bacteria. So try to think about what that is. That was Yersinia enterocolitica, all right? Now, it's dysentery, it's invasive. We're gonna get blood and pus in the stools, like we talked about for invasive diarrhea, okay? What form is transmitted, okay? You acquire entamoeba histolytica by fecal oral contamination, and you're typically ingesting the cyst form of the organism, okay, by fecal oral transmission on contaminated fruits and vegetables or contaminated water, for example. How are you going to diagnose entamoeba? You're going to look for the trophozoites or the cysts in the stool. And they're actually fairly easy to pick up because the nuclei have a very sharp central karyosome and little chromatin spikes. And sometimes you can actually identify red cells inside of the organism. So that tends to be very diagnostic for entamoeba. The treatment for entamoeba histolytica is metronidazole followed by iodoquinol, all right? And we do see infections from entamoeba in the US, but not as commonly as you're going to see entamoeba infections in developing countries. So we do see this in the US. It's probably not one of the leading types of parasitic infections, though, that we do see in the United States. All right, so we're going to move down the list. We're going to talk about Giardia, which in the United States is the most prevalent enteric parasite. You can almost probably write the vignette yourself. You've got individuals that go camping and hiking, and they drink untreated stream water. They then develop a malodorous diarrhea with a lot of fat in the stool, so there's steatorrhea. And the reason is because Giardia causes malabsorption. So I'm telling you, you have a foul-smelling diarrhea, and you're all sitting at home going, is she kidding? Foul-smelling diarrhea? Like that sets it apart from anything? So let me try to explain. I'm going to give you that. Yeah, diarrhea, you probably can't find any of it that smells good. But this is just like, bam, it's like a notch above foul smelling. Why? Because in addition to the normal stench that you're going to have, you have malabsorption. So your intake of food is simply rotting in your gut. So when you alleviate yourself, when it comes out, it's going to be really, really foul smelling. So that's why they even mention that here, because of the malabsorption. Now, this is a relatively poor picture of Giardia, but you know, students tell us all the time the images aren't very good on the board. So if you see an image like that, at least you should be able to identify it as Giardia. The image though here is what I would consider a very good picture of Giardia. You have to be ready for an image of Giardia. You know, they're going to ask you about it, and it's very ripe for the picking for them to show you a picture. Now, I actually think Giardia is kind of cute. If you look at this guy here, look, he's got two eyes, he's got a mouth, and he permed his hair, and he's smiling for the picture. Now, you don't get much cuter than that for an agent that causes malodorous diarrhea, okay? So how are you going to acquire this? Again, fecal oral transmission is key. And in the vignette, they're going to mention that untreated stream water, somebody with a recent history of camping or hiking and drinking that untreated stream water. We're going to diagnose it by finding these organisms in the stool. And you can see if you get a good image, good microscopy, it is very easy to detect. And you're going to treat Giardia with metronidazole. All right, so Giardia is going to be one of those high yield parasites that you're very likely to see on the boards. In addition to understanding the clinical symptoms, you also want to be able to recognize the image. All right, the next pathogen in our table here is Cryptosporidium. And Cryptosporidium shows up as a persistent, watery diarrhea in AIDS patients, okay? And it's very, very common in AIDS patients, very difficult to treat. So we have transient diarrhea, usually in immune compromised or AIDS patients. And the transmission is cysts from undercooked meat and then contaminated water. This is an example of a cyst. Now, this is an acid-fast staining, okay, of a cyst, but Cryptosporidium are actually acid-fast. The cysts are actually acid-fast. So that's one of the things that they're probably going to mention. So diagnosis, you have a 32-year-old patient who is HIV positive that comes to the clinic complaining of persistent watery diarrhea. You do a stool sample, you find acid-fast oocysts in the stool, okay? In that case, it's likely Cryptosporidium, all right? It's not the only thing it can be, and I'm gonna tell you some other things that it might be, but Cryptosporidium is probably the most common. Okay, as far as treatment in a healthy patient, it's usually self-limiting. In immune-compromised patients, though, this is very difficult to eradicate, very difficult to treat, okay? Now, we've talked about Cryptosporidium. Isospora bellii is another parasitic agent that causes persistent watery diarrhea in AIDS patients. Again, fecal.
I thought this one also had a section on B. coli, but anyway, I'm going to include this link for you guys.
But these are spherical.
Oops. All right, so let's stop. Let's.
Oh, we're going to get a discharge, microsporidia. And we're going to put microsporidia.
I have one more here that we did talk about, trichomoniasis. Let's look at this one.
Fairly low down on the list, okay? So again, we've got acid-fast, persistent diarrhea, but these, in addition to staining
the next one she's going to talk about is
also stain
trichomoniasis.
Okay, so Cryptosporidium and Isospora, those are your first two that you want to think about, and then Cyclospora and Microsporidia kind of down there at the bottom. All right, so those two are pretty low yield for step one. Trichomonas vaginalis is actually really high yield for the boards. It's going to be one of the things that you're going to have on your differential for sexually transmitted disease. Trichomonas, and we've talked about this a little bit before when we were comparing it between bacterial vaginosis. You're going to get a discharge, but in this case, it's going to be a frothy yellow-green discharge. That's different from bacterial vaginosis, where you have a thin grayish discharge. But again, it's malodorous, okay? And it's always described as a fish-like odor. So that's going to be one of the big clues. In addition, with trichomonas, you have a lot of inflammation that you can see here. So sometimes this is called strawberry vagina because of all of the erythema associated with that particular infection. Now, how is it transmitted? It's transmitted sexual, but what should scare you is this third bullet point, fomites. What's a fomite? It's an inanimate object, not person to person. So if you share a towel with somebody that has trichomonas, you can get trichomonas. So in that particular instance, you have to be pretty careful. Diagnosis are motile trophozoites, and here's an image of one. It's got this nice big nucleus, and again, it's got a little hairdo. Those are the flagella, okay? If they show you an image of that, you're expected to know what that is, or if they mention motile trophozoites in a wet mount, okay? In old school, when doctors used to have microscopes in their office, they used to suspect trichomonas. They would just do a little swab and they would bring it to their microscope, and you'd actually see these little guys kind of swimming all around underneath the microscope. So it's fairly easy to diagnose, but you have to recognize the image and or the description.
All right, so let's stop there. So I always like to sort of interject with another video or illustration to bring home the things that we've been talking about. It's always kind of good to hear it, from another source, if you will. I'm trying to share my screen. Share. Okay. See, B. coli. All right, so yes, so again, the same with ingestion of contaminated foods or water, and it's going to follow that same cyst trophozoite life cycle between encystment and excistation, okay, and the release of the cyst in our feces. Clinical symptoms of B. coli, abdominal pain or tenderness, nausea, anorexia, watery stools with blood or pus, ulcerations of the intestinal mucosa. All right. Most cases with a B. coli infection are going to be asymptomatic again. So basically, as long as you're healthy, you may or may not present these clinical symptoms. We talked a little bit about diagnosis, particularly for protozoan infections. We're going to have a microscopic examination of feces, right? So you're going to have to collect the stool. If we can get a formed stool, we're probably going to see those cysts being present. We probably will need those watery diarrheal stool for us to find active trophozoites. Okay. And I mentioned showers. As you're working on your extension assignments, you may see mention of these organisms appearing in showers. And so one stool sample collection may not be enough for a definitive diagnosis. So several stool samples might be necessary in order to get a true diagnosis because of the showering effect, where they may or may not be present at any given time because they appear in showers. Okay. You do have an extension assignment. Ignore the date. This is for today. You can submit this by midnight. You are describing the morphological stages of protozoan parasites that enter humans via ingestion. All right, and then you're going to discuss the possible clinical manifestations of an infection with Entamoeba histolyticus. So we talked about that in great detail today. I am going to upload these slides eventually, but for right now, you can use your notes and any other resources that you need to answer these questions. You will submit them in Blackboard. Don't email them to me. There is going to be a tab available for you in Blackboard to submit this by midnight. So go ahead and grab a screen snap of this, or however you would like to do it. Again, this is due by midnight this evening. What questions do you all have for me?